Thank you so much to this sounds really loud. Am I really loud? Thank you so much to the organizers for the invitation to speak with you today about my research. This is my first time in Lithuania and it is absolutely beautiful. So I'm so delighted to be here and to see your campus and to also get to see a little bit of the city. It's been a real treat and I've been looking forward to it. So thank you so much for the invitation. So this is just a pictorial of this is my daughter and my husband sneaking in the back, not realizing about the traffic in the city. So anyway, this is a pictorial of the obesity epidemic, and represented the larger the square, the higher the rate of obesity. And obesity is now so common that it's replacing undernutrition and infectious disease as the most significant contributor to ill health. And that's what I really want to impress upon that point, is that the reason to study this is because of the effect on health, that obesity increases the risk for a wide range of conditions, not just the ones you might be aware of, like type 2 diabetes and cardiovascular disease and cancer, but also a whole host of other conditions. In fact, it's like over 700 medical conditions that have an increased risk due to obesity. So that's the reason why it matters to try and understand it. And the United States is leading with the highest rate of obesity. Now, many of you weren't around in 1985, but I was, and it looked like this in the United States. So what does that mean? This means that obesity rates were very low. And this is actually even just, this is just overweight. So overweight rates are now over 65%, but you can see back in 1985, they were less than 10%. So the darker the color, the higher the rate of obesity. And what I want to show you is I'm just going to go through year by year, and you can see the year you were born and what America looked like then. But the reason why I'm showing this to you is to just illustrate that obesity is a modern epidemic. It only happened recently. It's happened in my lifetime. So we've gone from a state where there wasn't obesity to now there is, where it's very, very prevalent. So this is just an example of how the landscape of the country is changing, where people are getting more and more and more overweight. And you can see also that the states that are associated with more fried food, which is in the South, those are the ones that are increasing their rates of obesity faster, whereas states like Colorado and New Hampshire, which are more active states, they are staying at lower rates, but you can see that it's continuing. And now, this is from 2022, now they had to introduce even new colors because the rates are so high. So this is just to illustrate how quickly obesity went from not being present to being incredibly prevalent. And this is what it looks like in Europe. And Lithuania, the laser pointer unfortunately doesn't work. Oh, there it is. Wait, hello. There's Lithuania just there. So you can see that you are also quite prevalent in the rates amongst Europe, but the UK is higher. And so again, this is just showing how it's increasing with time. So how is body weight managed? It might seem counterintuitive, but it's managed in the brain. And that's because the brain is the master controller of food intake and energy expenditure. So that's, our brain is our command center. And that's where, that is where all of the obesity medicines primarily are targeting, is the brain, because that's where food intake is controlled. So I'm going to run through in today's talk a few different medicines. One is diphenfluramine, lorcaserin, setmelanotide, which is currently prescribed, semaglutide, which is currently prescribed, and tirzepatide. So to start with, and you'll see a little menu on the left-hand side, which is just telling you which drug I am talking about. So, how did serotonin become a target for an obesity therapy? It was discovered in 1948 by Rappert, Green, and Page. It was found in the serum, so it was named serotonin because it caused constriction, and it was found in the serum, so it was serotonin. And in the 1970s, researchers realized that brain serotonin is inversely related to food intake. So higher levels of brain serotonin led to reductions in food intake. So this is supposed to be me. A colleague did that for me at a conference a couple months ago. So the question I asked when I was a student is, can serotonin medications be used to improve obesity? So given that inverse relationship. And the answer was, yes, they can. And in fact, people were quite excited about it. And Redux was a medicine. And the question was, is this going to solve the emerging obesity epidemic that was only just starting? And how does it work? So what diphenfluramine does is it stimulates the release of serotonin and blocks its reuptake. So the serotonin cell is the yellow one, and you can see what diphenfluramine is doing is it is coming into the serotonergic cell, and it's stimulating the release of serotonin. Those are the little yellow balls. And then it's also so that it's available to act at its receptor, and it's also blocking its reuptake. So it's able to act longer at its receptor. So my first postdoc that I did, I investigated how that drug produces its beneficial effects on body weight, and that is by acting through the brain melanocortin pathways. And this is it's CFOS, which shows neuronal activation, and the brown is the POMC cell. I was very proud of that microscope picture, which looks very old school now, 20 years later, but I'm still putting it there because I'm still proud of it. So what's the brain melanocortin system? It's a really, really cool system. We don't have many of these in our bodies, but it produces both an agonist and an inverse agonist or antagonist. So that's quite unusual. Usually when we make hormones, they just do one hormone does one thing, but our bodies make the agonist and an inverse agonist or antagonist. And they work together. They compete for action at two different receptors in the brain, the melanocortin-3 receptor and the melanocortin-4 receptor. So pro-opiomelanocortin, you probably might be familiar with it because you probably know beta-endorphin. That's made by POMC. So that's one of the POMC peptides. Also ACTH, you probably know ACTH, stress hormone. That's also made by POMC. But the POMC peptides that are involved in appetite are the ones that are produced in the brain, and they are alpha and beta and gamma MSH. And they compete for action at these melanocortin 3 and 4 receptors with agouti-related protein, which is a really, really potent stimulator of food intake. If you give a mouse AGRP, just one dose, that mouse will still be overeating 2 days later. So it is a potent hunger stimulator. And if the POMC peptides act at the melanocortin receptors, then they turn off hunger because they're blocking the receptor. They're occupying the receptor, so AGRP can't act there, and they're also promoting satiety. So I came up with this hypothesis back when I was a postdoc, which is the way that diphenfluramine works, which is the picture I showed you. is by acting to stimulate serotonin to act at a serotonin receptor on POMC neurons to promote satiety, which then stimulates the release of alpha MSH, which can then act at a melanocortin receptor. So that was the hypothesis. And yay, it worked. Yay, right? So that's all good. And so, where's all my money? Nope, because it was withdrawn from the clinic. So it led to valvular complications in some patients. And so it was withdrawn from clinical use. So the question, so can 5-HT medications, can serotonergic medications improve obesity? Yes, they can. But the challenge then became to separate the beneficial effects from the side effects. So the question is which serotonin receptor is mediating the therapeutic benefit? Now, this probably doesn't even seem that complicated, but there are at least 14 different serotonin receptors, and they're grouped into 7 different families based on sequence homology and effector pathways. And when my former boss, lots of people just want to stay far away from serotonin because it is so complicated. But I love it. And in particular, the receptor that I was interested in is the serotonin 2C receptor, which is part of the serotonin 2 family, which is GQ coupled. And the reason why I was super interested in it is because my boss at the time, the boss that I went to work with, David Julius, he developed a serotonin 2C receptor knockout mouse, and it's the only receptor of all the serotonin receptors that when it's knocked out, it produces a hyperphagia and obesity phenotype. So what that tells us is that that is the receptor that serotonin is acting through to produce its effects on food intake and body weight. And to cut a long story short, because we don't have all day, we only have an hour, this did lead to a medication to treat obesity, and that was called lorcaserin, and that was the first new obesity drug in 12 years that was approved by the U.S. federal FDA. So the question that we were interested in pursuing is whether or not lorcaserin acts the same way that diphenfluramine acts through this melanocortin circuit. So as lorcaserin, you don't have to worry about stimulating endogenous serotonin, which is what diphenfluramine did, because that was then endogenous serotonin acting at off-target receptors in heart tissue. So now this is an agonist that's acting only at the serotonin 2C receptors, which are expressed only in the brain. And so the question is are they expressed on POMC neurons, and is the way that they're working through this melanocortin circuit that we discovered for diphenfluramine? So to answer that question, we used a very simple technique. We just used a knockout. So we used mice that didn't make POMC only in the brain, because as I mentioned, POMC is involved in other things as a prohormone. So we had this POMC mouse where POMC is not produced in the brain. And then we have mice where the serotonin 2C receptors, the POMC neurons that express the serotonin 2C receptors, those POMC neurons are not affected. So in the gray line, there's no brain POMC, and in the yellow line, POMC exists, is still produced, but only in the cells that express the serotonin 2C receptor. So what did we find? We found that lorcaserin requires these neurons to produce its therapeutic effect. So if it's a wild-type mouse that has functioning POMC, the drug produces the expected 40% reduction in food intake. Likewise, control mice that are just serotonin 2C receptor Cre mice, they also are responding to the drug. But if the mice don't make POMC, so only in the brain, then the drug is no longer effective. And if we allow POMC production only in the subset of cells that express the serotonin 2C receptor. Now this tells us that then the drug works again. So this tells us not only like specifically which neurons, as a class, but also which subset. So, because they're not every POMC cell has a serotonin 2C receptor on it. Some of them have leptin receptors, some of them have ghrelin receptors, some of them have GLP-1 receptors. So the ones that have the serotonin 2C receptors are the ones that are mediating lorcaserin's therapeutic benefit. So I mentioned, now let's just go to the second order neurons. So I mentioned that POMC and AGRP compete for action at the melanocortin 3 and 4 receptor. We suspected that it was the melanocortin 4 receptor that was the important target of this subset of POMC neurons. And to investigate this, we did the same thing. We used genetics, and now I'm just going to show you a simple experiment where we used mice that made melanocortin 4 receptors and mice that didn't make melanocortin 4 receptors. And we gave them lorcaserin. And as you can see that the wild type animals responded to lorcaserin by reducing their food intake. But if mice didn't have this second order receptor, the melanocortin 4 receptor, then they did not respond. So it tells us the circuit. And so this is just the model again. So the way that diphenfluramine is working is here at these yellow cells, the serotonin cells themselves, which are acting through the serotonin 2C receptor. I didn't show you data related to that, but we discovered that as well. And then lorcaserin bypasses the serotonin neurons and is acting at the serotonin 2C receptors on POMC neurons, which is then promoting the release of alpha MSH, which is specifically acting at the melanocortin 4 receptor. So this part of the circuit has also led to an obesity medicine. So this is setmelanotide, which is a melanocortin 4 receptor agonist. This medicine you might be less familiar with, and that's because it's only used to prescribe for genetic forms of obesity, like patients who don't make POMC or Prader-Willi syndrome and leptin receptor deficiency. Those are the genetic forms of obesity that are used with setmelanotide. So I just put this slide up because this was a crazy idea that I had when I was a postdoc. Now, this was, back in the early 2000s. What we know is that weight loss improves insulin sensitivity in obese individuals with insulin resistance, and that weight gain predisposes to type 2 diabetes. It's this, it's a linear model. It's a sequential model. Makes perfect sense. But I just had this idea that maybe lorcaserin could also be used to treat type 2 diabetes without weight loss, also through action at POMC neurons. And I persuaded the American Diabetes Association to fund this, and that's what's in the podcast, because it ended up being right, and it also improves type 2 diabetes in patients. Now, it's not currently used as a treatment, but the reason why this seemed like a very strange idea is because currently all medicines for type 2 diabetes act at the organs in the periphery. And so I remember colleagues saying to me, the body is very efficient. Why would it go all the way up to the brain to come all the way back down again? So it doesn't make any sense. The receptors are there. Drugs would not. That's not gonna work out. And if it does work out, it's because of weight loss. Well, I'm not going to show you any data related to this, but it turns out it was right. And if you gave a mouse or a person a single dose of the drug without any chance for weight loss, you saw improvements in insulin sensitivity and an increase in glucose disposal and reduction in reduction in hepatic glucose production. So this was seemed quite revolutionary at the time. Now, you guys are students, and it'll be in your textbooks that the brain can control blood glucose. But 20 years ago, it seemed like a crazy idea. Now, when you think about it, you think, well, it makes total sense. I started the talk by saying that the brain is the control center for the entire body. So, of course, it makes sense that the brain is involved. But at the time, it did not seem like a logical idea. So I mention that just to say that if you've got a crazy idea, go for it. And if people tell you it's crazy, if you have some data to support it, still go for it, because you don't have to just follow the same path that someone else has created already. You can create your own path. Right, so this is just on with the summary. So now we've got 4 different drugs that are all using the same circuit to produce their therapeutic benefit. So what about GLP-1 receptors? Do they fit into this brain circuit as well, or do they have their own brain circuit? And that's one of the things that we're investigating now. But I would say at the moment, there are 3 important questions in GLP-1 receptor obesity medicines. And that is the quality of weight loss. What are the drugs doing to body composition? The other question is, what happens when the drugs are withdrawn? Will body weight be regained? And is there any way to to combat that? And the third question is, where are they acting in the brain to produce this effect? So GLP-1 receptor agonists are also used to treat type 2 diabetes, and that therapeutic mechanism is via acting, as I mentioned already, directly at the peripheral organs. So, and the effect on food intake is via the brain. So starting with the first question, the reason why we're using mice, yes, I know there are lots of patients that are on this. So why are we using mice? Why don't we just go and do a patient study? It's because I can't persuade anyone to let me take their liver out and weigh it. Oops, sorry. Or to take their muscles out and weigh them, or to take their fat out and weigh them. So we're using the mouse because we can actually answer the question, because there's a lot of debate in the field, which is, are the drugs even affecting muscle, or is it all just down to improving the fatty liver? So we're able to pull the tissues out and to do histology and to weigh them. So just looking at body composition, what we're looking at when we're looking at fat-free mass, which is something we know is reduced in patients, we're looking at bone, liver, and muscle, and we're looking at body fat, we're looking at the different depots. So we know that body fat that's, around the stomach isn't great, but body fat that is on the hips and buttocks, that's not so bad. Brown fat, there, is where energy is being used. So is it affecting all the different fat pads the same, or are there special fat pads that are being particularly targeted? So Shuin did these studies. So we had mice on a high-fat diet for 6 months, and then we gave them semaglutide or Wegovy for, in this study, 10 days, and we looked at body composition. And so in a mouse, 10 days is long enough to see the full therapeutic benefit of about 10% weight loss. And in this graph, I'm showing you the drug-treated mice compared to control-treated mice. And in the other graph, I'm showing you the mouse compared to itself. So you can see that whether you compare the mouse to mice that just got the control treatment, or you compare them to themselves, there's a significant reduction in body weight. And this is accounted for by a significant reduction in fat mass and fat-free mass, which is what we would expect. And then the next question is, what type of fat? So now we're taking the fat pads out and we're weighing them. So if you look at all the fat pads combined, you see a significant reduction. If you look at subcutaneous fat, there's a significant reduction. And the RP WAT, there's a significant reduction. And the gonadal fat, there's a significant reduction. And if you look at the brown fat, there's a significant reduction. So the drug is significantly reducing all different types of body fat. It's not preferentially targeting one versus another. Then if you look at liver, I did go to a conference recently where somebody stood up and said that the entire effect on fat-free mass is accounted for by liver weight. So we looked at the liver, I agree. There is also a significant reduction in liver, and this is consistent with a previous study where they used twice the dose of drug that we used, and also they gave it for 12 weeks. We only gave it for 10 days. And same idea, we saw a significant reduction in liver weight and also a reduction in liver lipids. So the fat in the liver was also reduced. But then we took out the different muscles and weighed those, and those were also, the weight of the muscles was also significantly reduced. And we looked at different muscles. The gastrocnemius muscle is the largest muscle, so that one was the easiest to weigh. There was a significant reduction in that one. And the other three didn't reach statistical significance, and they're very, very small. So I would say I don't think that it's specific to gastrocnemius, but that's the one that was statistically significant. So another question in patients is whether or not there is a change in muscle function. So if you have a reduction in muscle mass, does that translate into a reduction in muscle function? Because you don't really want that. You don't want to be weaker. So one of the ways that we do it in mice and we do it in patients is using this test called grip strength. And what you can see is that it is not changed. Now, if you compare the mouse to itself, it's slightly lower, but it didn't reach statistical significance. So there was a paper that came out recently in patients where they did a muscle biopsy, and what they said is that the reduction in muscle weight is due to pulling lipids out of the muscle. Well, that would be quite good to, take the fat out of muscle, but I think the degree of muscle loss suggests that it's unlikely to be the only explanation. But these authors were suggesting that that's why you wouldn't see a difference in grip strength or muscle function is because it's not because the weight that your reduction is due to fewer lipids. There's been a lot of interest right now in the effect of GLP-1 receptor agonists on bone. So is it reducing bone mineral content and density? In 10 days of treatment, we did not see an effect on bone. And again, we're using the same sorts of ways to assess bone that we would be using in patients. So we did not see this. One of the papers in People that showed an effect was looking particularly at certain bones like hip bone. So what we might do now is reanalyze and look at specific areas of bone, but you can see that we're not really getting any indication of any effect on bone. So just to summarize that first study, 10 days of semaglutide treatment reduced all different types of body fat, reduced liver weight, reduced muscle mass but not function, and did not appear to have an effect on bone. And we're now looking at genes and proteins in these different tissues. So moving on to the second question is what happens to body composition when the agonists are withdrawn? And this is a really important question. So because many patients don't want to stay on GLP-1 receptor agonists their entire lives. It may be that as we're doing this research that is something that is necessary, but certainly a really active area of research is how do you maintain the weight loss when the drugs are withdrawn. So to do this, we gave the drug for 2 weeks and measured body composition before and after. So this is a little bit of what you would expect, what we're seeing in patients, and that is that during the drug period, there's a significant reduction in body weight, which returns after the drug is withdrawn. Not a super surprise. If you have hypertension, You take a drug for your blood pressure, you take the drug away, the hypertension comes back. So it's not a super surprise that the body weight effect goes away when you take the drug away. This was, and this is something that has just been reported in a meta-analysis of earlier this year. But this is something that concerned us because when we looked at body composition, there was more fat mass than when the mice started on drug. And this is the other concerning thing, is that the muscle mass, the fat-free mass did not return. So the mice, after they took the drug, the body weight resumed, but they had more body fat and the lean mass did not return. So this is something that we thought, how can we stop this? How can we prevent this? Can we prevent the muscle loss while retaining the fat mass loss? Is there a way to do it? So what Shuin did is he was very unhappy. He had to treat the mice every other day for 2 months. He was like, no. I'm like, come on, I did it when I was a PhD student. You got to do, or a postdoc. You know, this is research. This is science. You got to sweat for it. So anyway, every day for 2 months, and then you measure the body composition. So by slowing down the dosing, what we found is that we still got approximately the same degree of weight loss, but a significant effect on fat mass, but no loss of the fat-free mass, no loss of the lean mass. So this is quite literally $1 billion question. Lilly just paid $1 billion for a drug to prevent the muscle loss, and we did it too for free. So this is just by slowing it down. You don't have to add another drug. Just by slowing it down, we got the fat mass loss without the muscle loss. So you're welcome. No, so we'll see. We're really excited about this discovery. So we are just doing it in other conditions as well to see if this can be also demonstrated in patients. So that is the summary of studies 2 and 3 is just that there is a rebound weight gain. There is a rebound in fat mass that's higher than when the mice started. No recovery of the lean mass or the muscle mass. And by slowing it down, we were able to prevent this. So we're looking to now take this into the clinic. So the third and final question is, where do the drugs act to produce their therapeutic benefits? And, you know, are they acting in this same melanocortin circuit? I would really like the answer to be no. I want a new circuit. Come on, brain. You've got to, there can't just be one circuit. So we're hoping that it's not, that it's not the same. And so earlier research done by Novo Nordisk, they injected, they labeled the drug, looked at where it is in the brain, and you can see on the green slide that there are lots of GLP-1 receptors all in lots and lots of different brain regions, but GLP-1 is also produced in the brain. But where is the drug getting? It's getting to the pink areas, and that's actually only 4 brain regions, so not very many brain regions at all. And we're investigating those brain regions. And a real focus has been on this one on the bottom, which is the dorsal vagal complex, which is at the base of the brain just here. That's kind of a leaky blood-brain barrier, and that seems to be a really important area where the drug is working. So there's a recent paper by Amber where she investigated different subcompartments of that brain region and what they were doing. So in collaboration with Giles Yeo at Cambridge, we investigated, well, what are these GLP-1 receptors in this brain region, in this brainstem portion? And we found out that they are, a few of them are GABAergic. So a subpopulation of them are GABAergic. So the question is are those GABA neurons important in that in that DVC or subcompartment, nucleus of the solitary tract, are they important in the drug's effects? So we did some research on those, and we found out that those GABA neurons are really important in regulating food intake and regulating body weight. And then we went on to, which I don't have the time to show you, and then we went on to look to see whether or not the GLP-1 receptor agonists act at these neurons as well. So this was when Pablo was in my lab. He did this work, and he did an initial study with liraglutide. So this is just showing you the histology of the brain region. And he used this technique called chemogenetics, which some of you may or may not be familiar with, depending upon your background, but it's likely to be the future of medicine. So what is it? It's instead of having a drug act at a receptor that our own bodies make, it's an engineered receptor. So it's a receptor, a man-made receptor, and that binds to a man-made drug with high affinity, and we're able to put those receptors on wherever we want them to be, and then the idea is you wouldn't get side effects. And so, because you're only targeting specifically what you want to target. So we dropped in a GQ-coupled receptor, this designer GQ-coupled receptor, specifically on GABA neurons in this particular part of the brain. And then we turned those neurons off so they couldn't fire. And then we gave the animals liraglutide and found that it blocked liraglutide's initial effects on food intake, but not the longer term effects. And that makes total sense because this part of the brain is involved in initial effects of feeding, so up to 3 hours. So it'll be like the first few meals, but not like what you're eating all day long. So other parts of the brain tend to be involved in that other part. So it's more like short-term, it's more of a short-term feeding center. And then we did the same thing, we repeated it, and now this is Aurelia repeating it with semaglutide or Wegovy, and found similarly that it's necessary for a healthy chow food intake, but not yummy high-fat diet food intake. So the high-fat diet seems to be mediated by a different part of the brain. So this is some work that is unpublished and still ongoing. So sorry, he was giving a poster, but not here. So this is to show you what I also wasn't able to show you is more research that we did on GABA, but it does link in with the melanocortin system as well. So diphenfluramine is acting at the yellow serotonin cells, lorcaserin's acting at the green POMC cells, setmelanotide is acting at the purple melanocortin 4 receptor expressing cells. And one way that semaglutide is acting, just one of the ways, is through GLP-1 receptors on GABAergic neurons that are projecting to these potent hunger neurons, AGRP, and it's turning them off because GABA is an inhibitory neurotransmitter. And that's it. That's the whole talk. So to summarize, I talked about 4 different obesity medicines and how they act in the brain to produce their therapeutic benefit. We started with diphenfluramine, which stimulates the release of serotonin and blocks its reuptake, and it's acting at the melanocortin neurons to produce its therapeutic benefit. Serotonin 2C receptor agonist, lorcaserin, is also acting in that way. And setmelanotide is bypassing the whole serotonin system, going directly to the melanocortin 4 receptors to produce its benefit. GLP-1 receptor agonists are producing their benefit by acting in part through GABAergic neurons in the brainstem, which then project to AGRP neurons, which is the other side of that little scale that's balancing hunger and satiety, POMC and AGRP. And what we're interested in is trying to understand how these drugs produce their effects on body weight and the quality of weight loss, as well as how we can preserve muscle but still get the loss in body fat, and also how we can continue to sustain the weight loss. And that these are things that we are still actively pursuing. And then, of course, this is not something that was done in a day. This has been many, many years, pretty much my whole career of research. And this is the lab in different versions, the lab in Cambridge, early lab in Aberdeen, and the current lab in Aberdeen. And I tried to acknowledge people along the way. The most recent research has been funded by, that I presented, was funded by the Wellcome Trust and the UK government. And I just want to thank all of you for your attention. And I'm happy to answer any questions. Thank you.
Lecture, and for opening and broadening our horizons on the very relevant issue. This is obesity. And maybe I would like to ask everyone in the room, maybe you have some questions. Maybe you are, I don't know, keen in some very, very simple things which professor would like to answer. Okay.
Hello, thank you for this interesting topic you're presenting today. I would like to ask you a few things. If I understood correctly, so dividing the doses takes off the table the reduced lean mass loss, yeah?
Mm.
So you divided the doses throughout the week or so?
Yeah,
Elaborate on that.
So I think that's an important point, one that we don't have the answer to yet, which is my assumption. What we did is we used the therapeutic dose. We didn't divide it in half. We used the therapeutic dose, and we gave it less frequently. So it would be analogous to a patient taking an injection once every 2 weeks instead of once every week, but still using the therapeutic dose. Would it work exactly the same if you have the dose? And my guess would be no. And that's because I think probably you need the therapeutic dose to get the reduction in food intake and the reduction in body weight. But then the body, before it has time to recover from this, then you give another dose. But it's slowing down the weight loss, so the body isn't panicking and taking calories or taking energy from the muscle. It's getting them from food. So I think that's the point. Is I don't think that if you halved the dose that it would work. But we haven't done it yet.
But have you considered, for example, if you ensure the sufficient protein intake during that, maybe this lean mass also can be preserved?
That is something that people are investigating, but I think the answer so far to that seems to be that it might slow down or reduce the degree of muscle that's lost, but not take it away completely. So for patients, it is important who would have the muscle loss. I think it's important to have protein and to do resistance training to try and prevent it from being lost. Those are current strategies, but they're still seeing muscle loss. So this is why we're looking at another strategy.
Another question. So this was only with this single agonist, yeah?
Mm-hmm.
And what do you think about these double and triple agonists, which are now very popular?
Yeah, I think it would probably be the same, but we haven't done that. The difference is that, so there are two strange differences. One is that tirzepatide or Mounjaro doesn't produce as much lean mass loss as semaglutide. It still does, but just a little bit less. So it seems to spare it a little bit. that's a little bit counterintuitive because it produces more weight loss. So you would expect more muscle loss, not less. So it's a little bit better at sparing it, but it'll be the same.
And one personal question, if you don't mind. Would you consider taking these drugs yourself?
Are you calling me obese?
No, no. If
No,
if
I'm
the,
just kidding.
if
I'm just
yeah,
kidding.
yeah, yeah, yeah.
Yes. I think I think it's very important to point out the reason this is medicine. This is not a vitamin. It's a medicine. Medicine should only be taken for medical reasons. So in the UK, these medicines are only prescribed for people whose body weight is producing a condition, a disease like type 2 diabetes or cardiovascular disease, etc. So without a medical reason, you would not be allowed to get the drugs on the NHS. So I think that no, I don't think everyone should take them regardless of body weight. I think it needs to be obesity, and it needs to be obesity that is related to a health complication.
Yeah, but what I mean that the elephant is in the room. Yeah, they are so popular.
I see the elephant. I see the elephant.
It flooded via all kind of markets, the Europe and the world. Yeah, so you don't
Yeah.
need to go to NHS or whatever to
Yeah.
get these drugs here now. So and these people are
Are
taking
you
them.
selling them later?
No, no, no,
No,
no.
I'm
I'm
just
just
kidding.
interested in your opinion about
It's supposed
what's
to be, I mean,
long-term damage. You know, that's what I'm getting where to. Yeah.
it is supposed to be prescriptions. I don't know what the rule is here, but the drugs are marketed to treat obesity. If you don't have obesity, you shouldn't be getting the drugs. So if people are allowing, prescribing, if doctors are prescribing, it would be unethical to prescribe a drug for something that is a condition that the person doesn't have. So it's against the legislation and regulation. I'm not so naive to think that people don't get the drugs in another way, but I mean, we have to just work within the legal system. So I can't comment on people using the drug outside of what it's supposed to be used for.
But you see some kind of dangerous long-term effects in these GLP-1 agonists.
I think that remains to be seen. I think what people don't fully realize is that drugs in this class of drugs have been prescribed for 40 years. We know what they do in people because they've been prescribed for 40 years. The formulations are getting better and better, but initially it all comes down to how quickly the molecule is being broken down. Our own hormone is broken down within minutes, so within like 2 minutes, GLP-1 is broken down. The first versions of the medicines were broken down quite rapidly, so they had to be injected twice a day. Then the next version was once a day. Now the current versions are once a week. There are ones in clinical trials right now which are once a month. There's also drug discovery already, and drugs already on the market that are oral tablets instead of injectables, which are every day. So, but this class of drugs has been in human use for 40 years. We know pretty well what the class of drugs does to the body and the side effects. So I don't think there's like massive danger. I'm not super worried about unexpected side effects that would have catastrophic effects on health. The new medicines are pulling in other systems, so the things that are on the horizon. So we start with just GLP-1, acting at GLP-1 receptors, or drugs that are mimicking GLP-1. Tirzepatide is GLP-1 and GIP receptor agonist. We've got new drugs that are coming that add in amylin, new drugs that are coming that are adding in glucagon. So as we start adding in other things, now it's something that hasn't been around for 40 years, but just the ones that I would, initially, Wegovy, Ozempic, that class, just GLP-1 on its own, has been around for 40 years.
Thank you for your answers. I have one more question. Maybe someone wants to.
Thank you for your interesting questions.
The clinical, how looks like in the clinical settings. So these prescriptions, because in Lithuania, if I may, so in Lithuania we could prescribe only by prescription. So there are various situations how people get the drug, but in Lithuania we use only prescription, and so this is quite strict to get these medications. And of course, I agree with Laura that there are some adverse events of these medications, and some of them, I don't know, that it might be blindness. For in some cases it might be constipations, and people just withdraw because sometimes it takes more than 2 or 3 weeks. In the case it might be nausea. Some of the patients can't use it because of the nausea and vomiting. They are changing to another one. So, and it is, this is a drug, so it might have adverse events. So you need to be followed by the physician how you use the medication. Because, for example, I agree with Laura as well that we suggest to take the proteins at least from 1 to 1.5 grams of protein to 1 kilogram of the weight. It means a lot. If you start to calculate how you eat during the day, it's quite a lot of.
Especially if you're not feeling hungry or you're feeling
Because you
nauseous,
don't
it's
feel
quite
it.
hard
So
to,
that
yeah.
it might be the reason, but the very important one is the physical activity resistant one. So if you don't follow the conditions how you use the medication, it doesn't work or work in a proper way. So
I think there was a question.
There is many cases where a drug was developed that worked well in mice, but it didn't work in humans at all or had many side effects. So my question is, what is your personal opinion on doing research on mice and how well it translates to effects that we see in humans?
That's a really, really good question. And I think that it's one to always keep in mind that, to not do animal research unless it's necessary. But certainly animal research does provide value in considering the efficacy and the potential side effects of new medicines, and also understanding how medicines act. The ones that I've been interested in are ones that are really fundamental circuits that are the same in a human as they are in a dog, that they are in a cat, as they are in a rat, as they are in a mouse, and you can keep going down to different organisms. And those are the ones that I'm interested in, because anything that's eating to survive will be acting on, will need circuits to provide that impetus to go and seek out food and to consume it. So those are the ones that I tend to study. But of course, there are lots of things that animal research is completely unsuited, or situations where like humans don't have tails, so we wouldn't be able to do tail research in a human. So like you need to have a species that has something. So we're not all identical. But for sure, I would prefer to do safety research in a mouse than in lots of monkeys, for example. So I think when we have medicines to improve health, we just need to be very aware of the value of the research and make sure that it's really contributing, and that they're discrete, important questions that are being answered. So thanks for that.
Thank you.
thank you very much.
I think there was another question over here, but maybe that one got answered.
Oh. Thank you. So I wanted to ask about these side effects, because you said that first substance was withdrawn because of side effects for heart, probably.
Yeah.
And then that lorcaserin was also withdrawn. And
Yeah.
I didn't catch why it was withdrawn, but in conclusions it was said that it's not in use anymore.
Right.
And was there also some side effects? And my question is, what type of side effects? And can we somehow prevent occurrence of such side effects before we give medicine to patients?
Yeah, that's a really good question. And you listened quite carefully because I purposefully skipped over that because it's a little bit complicated, but now I will address it. So lorcaserin was withdrawn because of the cardiovascular effects. So those are the serotonin 2B receptors that are in the heart. So if a mouse or a person doesn't make a serotonin 2B receptor, then the heart doesn't develop normally, so it doesn't survive. So we know that the serotonin 2B receptors in the heart are really important for survival. So because the serotonin 2C receptor has a decent degree of sequence homology with a 2B receptor, when the FDA approved lorcaserin, they said to the drug company, you're going to need to follow patients all over the world for 5 years to make sure that it doesn't cause any sort of cardiovascular side effect. And the drug company was delighted when they unblinded everything to find that it didn't cause any cardiovascular side effects. And in fact, it improved type 2 diabetes, which was consistent with the research that we did in mice. So I was like, yay. Now, I mentioned at the beginning of the talk that obesity predisposes to cancer. So of course, in these patients, if you're following them for 5 years, a small percentage of them will develop cancer. And in this particular case, it was in the placebo-treated individuals, it was 7.2% of people developed some form of cancer. And in the drug-treated group, 7.6% developed some sort of cancer, no statistical significance related to that. So the FDA came back and said, we want you to follow longer in case the drug increases the risk of cancer. Well, the drug doesn't, they just did that. It doesn't increase the risk of cancer. Obesity does. This is not news. So the drug company said, no, we've just spent $1 gazillion following people all over the world for 5 years. We're not going to do that. And so then the press releases were lorcaserin causes cancer. It's not. So sometimes the press, doesn't really review things, but quite carefully and takes the wrong tagline. But now if you Google it, it says lorcaserin causes cancer. It doesn't. Never did. Obesity does. That's what causes cancer. So it's I think the real reason why, I don't know, I don't sit on the FDA and I don't work in the drug company and I never have either of those things, but I think the real reason why the drug company pulled it because they said we're not following patients for another 5 years. And I think they knew the GLP-1 receptor agonists were coming and they wouldn't be able to compete with that degree of weight loss because lorcaserin causes 5% weight loss and the GLP-1 receptor agonists currently produce 10 to 15% weight loss. So patients would be taking those medicines first anyway. So I think it just wasn't economically viable. Yeah.
Point about them trying to increase the half-lives, yeah?
Yeah.
Yeah, but then if the body works in pulsatile manner, no?
Yeah.
Majority of the hormonal systems, yeah, like GH or whatever, and GLP, as you said, super short half-life.
Right.
And then you deviate from these natural processes. And then I'm curious, is it because it's more profitable or because other reason,
It produces more weight loss.
Yeah, but why don't you develop, not you, whatever, hypothetical research, yeah, develop drugs to work in a pulsatile manner, yeah, like let's say glucose monitor, yeah. If you are hungry, then you press a button and you're not hungry anymore.
It's a good
for
idea.
some, and then you mimic the natural pulsatile systems.
You could be sitting on a gold mine there. All you need to do is just
We all are, maybe.
No, it's a good idea. But yeah, I mean, you could. I haven't heard anybody suggest that, so that's a good idea.
But my question is that don't you think that's where the side effects and interactions and health issues is coming from? Because of the contradiction with a normal pulsatile bodily functions, and then you are developing, not you, but whatever, person, drugs which has a half-life of weeks, whatever, no.
Yeah,
That's the problem.
I think the side effect of nausea is being mediated by a particular part of the brain called the area postrema that the drug needs to get into the brain, and so I think you're going to get the nausea no matter if you do pulsatile or prolonged. In fact, you might even have less chance of nausea if you do prolonged because the drug will leave the receptor and you'll start having other effects. I think it's the poster session now. I think we should maybe wrap up.
Yeah, thank you very much for your lecture. I think that this class of the medication, this is the future. And not only because of the obesity, because of the effects on neurocognitive function as well, due to addiction. Because we didn't have time, to talk about addiction, because it decreases addiction not only to the food, but addiction to smoking, to the alcohol, and so on. So this is a very broad type of the medications, and they work really very well if they used in the correct patients, and in correct way. So we need just to keep and to trust. To keep on the path of being trusted by the physicians, by the researchers, and take these medications.
Right. Well, thank you, everybody.
Thank you very much. Thank you, Professor.